So I'm Jasper. I'm going to be introducing the next keynote speaker, which is my honor. But I just wanted to give you a brief look behind the scenes where you spend a year preparing a conference with spreadsheets and Gantt charts and lots of Zoom meetings, and at some point you kind of feel like you're there. You kind of feel like, we're on top of things. And then yesterday, we hear that Anna's plane had to turn around because of a mechanical failure. So lots of frantic emails and everyone's stressed out for a little bit. So we feel very, very lucky that we got to see you here in person and you get to present here. So Professor Anna Schapiro is associate professor at the University of Pennsylvania. Before that, she did her undergrad at Stanford on symbolic systems, a PhD at Princeton, a postdoc at Harvard. She's won a ton of grants and awards, such as the Psych Science Rising Star Award, the CNS Young Investigator Award, and her work has contributed greatly to the study of learning, how we build our knowledge of the world, and the things that we experience.
Great, can you hear me? Yeah, it's working. Hi, thank you so much for having me. I'm really happy to have made it here. Yeah, this was our flight path. Me and a bunch of students from Penn. At least one of them came straight from the airport this morning to do his poster. So shout out to Jake. And I got to sleep for a couple hours in the hotel. So hopefully I'm awake enough to answer your questions today. So thank you so much to the organizers for inviting me. I'm really, really excited to be here and to share our lab's work with this group. So today I'm going to ask this question: how do we learn across timescales? So we need to learn both slowly over many hundreds or thousands of learning opportunities, and that can play out over weeks, months, years, but we also need to learn quickly, sometimes within just a few exposures. So if you're visiting Amsterdam for the first time, you need to learn very quickly to look out for bikers and avoid the bike lane when you're walking on the streets. And to the extent that you're trying to learn that new information in the same neural populations that represent your existing related knowledge, there's this danger of overwriting that existing knowledge. So if I'm going to try to use the same neurons to represent navigation in Amsterdam and navigation in Philadelphia, I'm going to have to contend with this interference issue. And I want to be able to go back to my Philadelphia optimized street navigation where nobody's paying any attention to the bikers. So this is, of course, a difficult and kind of infamous problem for neural networks that use distributed representations. And so how do we do this? So one proposal for how we do this, of course, was the complementary learning systems theory, which said, well, maybe the way that we learn at these two different timescales is that we have a region, the hippocampus, that will specialize in the rapid learning of individual new experiences. And it will use this pattern-separated neural code that will allow you to very quickly encode that new information and kind of separate the new experiences from one another to avoid interference and recall each of those individual experiences. And then the idea is that the hippocampus can do interleaved replay of these experiences offline, maybe especially during sleep, and that would allow the rest of the brain to slowly build up that kind of integrated, distributed representation that's going to ultimately represent all of your knowledge of the world. So the idea is to kind of turn that non-stationary distribution from the environment into a stationary training distribution by having the hippocampus do this interleaved replay for you. So the idea is you avoid the interference problem by not writing the information directly to that ultimate knowledge store, but instead storing it first in the hippocampus and then carefully integrating it offline. Okay, so that's well and good, and I think explains a lot about how these memory systems interact. But to solve the complete puzzle of how we learn across timescales, I think there's a few more really key questions that we're going to need to address. One question is, how do we learn across experiences quickly? So the complementary learning systems theory kind of told us how we learned individual snapshots of experiences, but it might take you a few experiences of almost getting hit by a biker to understand how to navigate the streets of Amsterdam. And so how do you integrate across those experiences quickly? And then where and when can learning occur directly in neocortex? So what kinds of information needs to go through that process of hippocampal storage and then replay for integration? And what kind of information might we be able to incorporate directly into those kind of like ultimate neocortical knowledge stores? And then finally, how does sleep and replay actually serve to integrate hippocampal and neocortical knowledge? So our lab is working on these 3 questions, and I'm going to give you some examples of what we're thinking about the answers to these questions. And just to preview the 2 key claims that I'm going to make, one is that the hippocampus is not homogeneous, and that part of the answer to this question of how we learn across timescales has to do with different anatomical structures within the hippocampus. So we propose that there are rapidly learned distributed representations, kind of like the ones out in neocortex, that complement the classic pattern-separated representations in the hippocampus. And then neocortex, of course, is not homogeneous either, and we are interested in the possibility that we can think of there being kind of a gradient of these properties increasing overlap or increase in the distributedness of the representations and increasing speed of learning as you go from early sensory areas up through medial temporal lobe cortex and into the hippocampus. Okay, so let's start with the first question. How do we learn across experiences quickly? So we got started thinking about the hippocampus as being potentially involved in this kind of learning when we were running these fMRI experiments where we saw that the hippocampus was sensitive to novel temporal statistics. So we were running experiments where participants saw these streams of novel images presented one at a time, but there was some hidden temporal structure governing the transitions in the sequence. So in this study, there were, let's see if I can point here, yeah. There were pairs of items that always occurred together. And we found that from before to after about an hour of exposure to these sequences, the hippocampus came to represent those associated items more similarly to one another. And it wasn't just the simple pairwise structure, it was also this more complex, maybe more naturalistic community structure. So here items in the same community came to be represented more similarly in the hippocampus. So this tells us that the hippocampus is it's sensitive to this timescale of statistical information, and that it seems to be representing the related experiences using this kind of overlapping neural code, which is not what you might have expected from the classic literature on the hippocampus. And then it's not just that the hippocampus is sensitive to this information, we think it's actually necessary for this learning. So our lab and others have found that patients with hippocampal damage don't express behavioral evidence for this kind of learning, so they're unable to detect the structure in these sequences, and that's not just for these like kind of shape stimuli, which we did try, but also for visual scenes and syllables and tones, and so we think it's a cross-modal kind of learning that this region is doing. Okay, but then so how do we think about the fact that we're seeing evidence for this kind of statistical learning, building these overlapping representations? How do we reconcile that with what we know about the hippocampus being very good at separating experiences for episodic memory? And to think about this, we've been working with this neural network model of the hippocampus that we call Seahorse that has properties and connectivity that implement many of the properties that we know of the actual hippocampus. So there is an entorhinal cortex layer that processes input and output from the model. It's set up as an autoencoder, so it's taking whatever pattern of activity is on this input layer and then trying to reproduce it on the output. And then we have 3 hidden layers that learn to do that mapping that correspond to 3 of the subfields of the hippocampus. So this dentate gyrus, CA3, and CA1. There's 2 pathways through this model, as in the actual hippocampus. There's this trisynaptic pathway and then a monosynaptic pathway. The trisynaptic pathway is the pathway that's famous for these sparse pattern-separated representations that we think are so important for episodic memory. So there's this very kind of specialized, sparse fanning connectivity into dentate gyrus that we think is taking input in entorhinal cortex, even if it's very overlapping, and projecting it to separate populations of neurons in dentate gyrus. But then there's this direct pathway from entorhinal cortex to CA1. And this pathway seems to have some different properties. So there's less extreme pattern separation in CA1, probably related to differences in its connectivity. And also, it has a slower learning rate than the trisynaptic pathway. So the trisynaptic pathway can learn as quickly as in one shot, whereas the monosynaptic pathway can learn on this time scale that we're talking about, but it seems to be more incremental. So we are very inspired by this kind of finding from the rodent literature that shows that if you specifically knock out the CA3 to CA1 connection, you can't do one-shot learning anymore, but you can still do incremental kinds of learning. Okay, so in our kind of original simulations with this model that I'm not going to go through here, we showed that these properties, so the increased overlap and the slower learning rate, make this monosynaptic pathway very well suited to the kinds of statistical learning that we had been looking at, whereas the trisynaptic pathway has properties that are very well suited to supporting episodic memory, as we knew from a long history of empirical and computational work. So the proposal is kind of that there's this microcosm of the complementary learning systems dynamic playing out within the hippocampus itself, where CA1 is acting kind of like a little piece of neocortex within the hippocampus, in the sense that it's doing relatively slower learning, which allows you to aggregate over more experiences, and it's using these more distributed representations, which is going to make it easier to see the relationships across your experiences and make it easier to generalize. And so, the idea is that it's not, CA1 is kind of it's a faster learning and not quite as distributed version of the neocortex that could allow us to do this kind of statistical learning on this time scale of minutes to hours. Okay, so if this model is right, then it kind of opens up a new learning strategy that we could consider for many forms of across-episode structure learning. So statistical learning is one example of a case where you need to integrate across your experiences to understand what's going on. But there's a lot of things like that, and we have been exploring whether this learning strategy might shed some light on other kinds of rapid learning. And one area that we're really interested in is new category or concept learning. This is another domain where you need to pick up on structure quickly across experiences. In this case, it's like, can you understand the category structure from experiencing different exemplars of a category? And we know that the hippocampus is involved in this kind of rapid new learning of category structure. So first we just thought we would apply the model to some canonical category learning tasks and see what happens. So here's how it does on the classic weather prediction task. So if you don't know this task, you see a set of these abstract cards, and you're supposed to predict sunshine or rain from the configuration of cards. And we know that amnesics are able to do this task to some extent, but they're not at the level of control. So it's not that the hippocampus is the only region involved in this task. In fact, there's plenty of evidence that other regions are involved, but it's contributing something, and we want to know, what might that contribution be. So here's the model's performance. So the green is like the regular intact version of the model. The orange is a version that only has access to its monosynaptic pathway, and then the purple is a version that only has access to its trisynaptic pathway. And the model performs similarly to people with a similar number of trials. And what you can see is that its ability to do this categorization is really dependent on the monosynaptic pathway. The trisynaptic pathway struggles with categorization. But if you query the model differently, so if you ask it to try to recognize particular configurations of cards that were seen during training, then the pattern flips, and you see that the trisynaptic pathway is much better at that than the monosynaptic pathway. So both of the pathways are learning something from the input, but they're learning, different aspects of the structure. I'm going to show you one more example simulation. So this is a paradigm that we've used a bunch in the lab. It'll come back again later in the talk, where participants learn about these categories of novel satellite objects, where satellites in the same category share most of their features, but they also have unique individuating features and a unique name. And we had found in one of our fMRI studies with this paradigm that CA1 represents this category structure. So the satellites from the same category represented as more similar than satellites from different categories, whereas that wasn't true in CA3 and NHIRIS. So here's the model performance on generalization. So after training on a certain number of trials, show it an exemplar it hasn't seen before and see if it can categorize. And we find that it does very well at this, and that the performance is, again, really driven by the monosynaptic pathway. But again, if you change what you're asking the model to do, and what you're really interested in is, can it differentiate between these exemplars in order to report on the unique individuating features? There's quite a lot of interference there, so this is the kind of situation where the trisynaptic pathway might be useful, and indeed the monosynaptic pathway really struggles with this amount of interference, and the trisynaptic pathway is able to support that unique feature memory. So it's not that the trisynaptic pathway is not useful for category learning. It is, to the extent that it's important to keep track of the exceptions and the kind of like arbitrary aspects of the structure. The internal representations of the model are what you would expect. So CA1 represents that category structure, whereas Dente and CA3 keep things separate, kind of like what we had seen in our fMRI data. So here, I want you to notice that the model, the intact version of the model is actually not doing as well as it could have done had it kind of listened just to the monosynaptic pathway. So it looks like there's a trade-off here in the performance of these two pathways depending on the task. And it would be nice if the hippocampus were kind of smarter and could say, well, if I'm in a generalization task, I really should be listening more to the monosynaptic pathway, but if I care about the unique features, I should be listening more to the trisynaptic pathway. So that this behavior where it's kind of averaging the two is not really optimized for this task. And we're very interested in the possibility that there could be interactions between hippocampus and prefrontal cortex regions, maybe especially the medial prefrontal cortex, that might allow the hippocampus to kind of deploy the right representation at the right time. So I wanted to highlight, I was just going to point out here that this trade-off is, you can see it as well in the weather prediction task, but it really is clear in this case. So I wanted to just briefly tell you about this new project we have led by Darius Singh, who's over there, who has a poster on this tomorrow, on this way of thinking about the interaction between a MPFC-like control structure in the hippocampus, and try to, explore whether this could make better, more adaptive use of these two different representations. So here's the version of Seahorse we're using. I wanted to point out that we finally have this implemented in PyTorch instead of Emergent, which is what we were using before. We're still using Emergent too, but we're really excited about the PyTorch implementation because people who are interested in downloading this model and playing with it, I think, are going to have an easier time now with this simpler version in PyTorch. And also, you can stick it on top of your favorite ConvNet. So here we're using CoordNet, so it's now a stimulus computable model instead of using the abstract hand-coded features that I had been showing you before. So we're very excited about this version of Seahorse. But then here, what Daria is doing is setting up a mixture of experts training situation where this medial prefrontal cortex region is going to learn to deploy these two different experts to the task that they might be most useful for. So here he has a simulation with Fashion MNIST where he has the model learning to categorize the images, but also learning the labels of the individual exemplars. And he finds the same trade-off that we had seen in the previous simulation. So the monosynaptic pathway is much better at the category task than the trisynaptic pathway, and vice versa for the exemplar task. But this mixture of experts version of the model is able to kind of take advantage of the expertise of each of these individual pathways and deploy it correctly so that you don't get this kind of trade-off averaging effect. So we are exploring whether this might help us explain kind of some of the empirical evidence for MPFC control of hippocampal representations and output, and help us think about kind of like adaptive use of these representations. Okay, so go find Daria tomorrow if you want to hear more about this. Okay, and then I said that, you know, category learning was one kind of task that we thought the monosynaptic pathway might be useful for beyond the statistical learning paradigm that we were originally thinking about. I want to give you one more example of a task where we think that this kind of learning strategy might be very relevant and important, at least under certain conditions. And this is also a task where there's been a lot of theorizing about how the hippocampus might contribute, so I think it's an interesting one for us to consider and see how our model relates to other models. Okay, so associative inference, you learn that A and B go together, you learn that B and C go together, and then there's some assessment of your ability to make that transitive link from A to C. We know that this task is dependent on the hippocampus from rodent lesion data, and it also comes up in our long history of fMRI experiments with this paradigm. So there have been different strategies proposed for how the hippocampus might solve associative inference. I'm going to focus on two of the strategies that I think map on to the two pathways of the hippocampus in the way that we've been talking about them. So this model is called Remerge from Kuhlmann and McClelland. This model kind of doubles down on the pattern-separated representations in the trisynaptic pathway and says we don't need to do any kind of, we don't need to build overlapping representations of anything, just stick with the pattern-separated representations of each of these individual experiences. But you can solve this inference through activity dynamics at test. So you keep A, B, and BC separate during encoding, but then at test, if you see item A, you can kind of circulate, you know, activity through this network to get to your BC and then out to your C. So you can get from A to C without ever having directly stored the relationship between A and C during encoding. That's very different than this strategy that I've been talking about, which is that, you know, we're going back and forth between studying A and B through interleaved learning and then building up this like overlapping distributed representation of A and C. So this is the way we're thinking about the monosynaptic pathway. It kind of starts out with some overlap between these items, and then as you notice that there's this B item in common between A and C, there's pressure for the model to pull those representations closer together, and then that makes the inference totally trivial because A and C are just sharing neurons by the end of that encoding process, and so it's very fast and automatic to do that inference. So, the prediction for the monosynaptic pathway learning strategy is that inference should be very fast and automatic, but dependent on having experienced these individual pairs in interleaved presentation order, because distributed representations, as we talked about earlier, are very sensitive to the order presentation of information. This kind of trisynaptic pathway strategy, it's slower at test, maybe more effortful. It involves having to go through these recurrent activity dynamics, but it's not dependent on the presentation order. It doesn't care how you were exposed to A, B, and BC. In fact, you could have just seen each one once. So we're going to try to take advantage of the differences in the characteristics of these two strategies to see if we can tease apart what might be happening. So this is a line of behavioral experiments led by Zi Zhou, who had people do an associative inference paradigm where the pairs in each triad were either presented in interleaved order, meaning you would see AB, BC, AB, BC, or in blocked order, meaning you would see all of the ABs for that triad before you would see any of the BCs. And then at the end of that sequence exposure, he did 2 tests. The first is a speeded recognition test, where we asked, were these 2 objects shown together as a pair during the study phase? So the answer here is no. So even though these 2 objects share this orange object in common, they weren't studied together, and so you would say no. And then after that, we do the classic explicit inference tests that people usually do with this task, where you show people an object and you ask which of these 2 objects below is associated with the object above, give them plenty of time to think through that and solve it however they would like. So we don't predict a difference between our conditions for the explicit inference test. If you've built up this directly overlapping representation of A and C, then this kind of trial is confusing, right? It might be hard to say that you did not study these 2 items together if you've built up this overlapping representation. So the prediction is if interleaved learning affords that kind of representation, you should be slower to say that you didn't study these 2 items together, or maybe you'll even false alarm and say that you studied those items together when you never saw them together. So here's the results for the explicit inference test. This is 3 versions of the experiment where we made minor tweaks. So you can think of these as replications. This last one was especially well powered, and there is really no difference between the conditions. So higher here would be advantage for interleaved, lower would be advantage for blocked. There's no difference between the conditions. But for the speeded recognition task, what we find is that there is this tendency to be slower to judge that the interleaved ACs were not studied together, which we think is consistent with this idea that the interleaved condition is allowing you to build this overlapping representation that makes that particular trial type confusing. And in 2 of the 3 versions of this, we actually found evidence for false alarms. So people were actually saying that they had studied those AC items together when they had not. So that's an example of where these putative distributed representations are not being useful. They're confusing you about what you had studied. But of course, we think that they should be useful for many kinds of things, and maybe the most important thing is generalization. And so we ran a version of the experiment where we did the same exposure, but then at the end, we told people that the A objects had some novel property, and we tested how likely they were to generalize that property to its indirectly associated C object. And we found that people were more likely to do that in the interleaved condition. And then finally, we thought it would be interesting to turn this, this paradigm into a statistical learning experiment. So instead of presenting the items 2 at a time in these demarcated pairs, we showed the items 1 at a time in a sequence. And the reason we are interested in this version is that this is the kind of situation that's very hard for a trisynaptic pathway-like learning strategy to handle. It doesn't know how to discretize into episodes, whereas the monosynaptic pathway is very good at handling this kind of noisy statistical input over time. And what we find in this version of the task is that even in the explicit inference, so when we give people plenty of time to think through the task however they want, they are now, there's now an advantage in the interleaved case. And if you break it down by condition, it's actually very striking. In the block condition, they're completely at chance. So if you had experienced these items in blocked order with this kind of statistical learning presentation, then you just cannot do the inference at test, whereas you can in the interleaved case. Okay, so far, I'm building up this story that we think is consistent with this possibility that the hippocampus is building these distributed representations in the monosynaptic pathway that complement the classic pattern-separated representations in the trisynaptic pathway. So we still think that those are important for episodic memory and maybe for some forms of inference in some situations, but that there might be this other thing going on as well. Okay, so far, we've been talking about rapid learning of completely novel structure, where your prior knowledge has no bearing whatsoever on the particulars of what you're about to learn, right? The structure is randomized across participants. There's no way you could have predicted what feature was going to go with what feature on those satellites, for example. But to address this next question, we want to ask whether these processes we're talking about might be different when you're learning in an environment that is actually representative of the distribution of your past experience. And of course, much of our learning occurs in that kind of setting, so it's very important to think about this. We're not often learning things that are completely novel. And here, we're still interested in the potential role of the hippocampus and region CA1, but when you are making small tweaks to your prior knowledge, you might expect that the regions that represent that prior knowledge would play an important role in this kind of learning. So we're also interested in looking out in neocortical areas. So here we're going to use the natural scenes data set, which is familiar to many of you, I'm sure. So this is a data set where 8 participants each completed at least 30 sessions of high-resolution 7T fMRI. They viewed 11,000 images of natural scenes. The images are tagged with 80 different possible objects like giraffe and table and bus. And these objects co-occur in the ways that you would expect based on your prior knowledge, because these are just real images. So we first built an object encoding model that allows us to predict voxel activity patterns for each of the individual 80 items. We did this by regressing each voxel's activity for each image against a vector indicating which objects were present in each image. And then we evaluate the encoding model on held out data, and we see successful predictions across a lot of the brain, certainly all visually responsive cortex, but also the hippocampus and medial temporal lobe cortex, which is reassuring, because it's actually not the case that we always see such nice object representations in the hippocampus. So we can get nice representations of these objects throughout the brain and up into the hippocampus. So these are the areas that have reliable representations of the objects in these scenes. And then within those areas, we wanted to evaluate which of these representations might have some semantic content to them. And the way we did this was to compare the similarity structure of the object representations to word-to-vec similarity structure. So it's a measure of semantic relatedness based on word associations and text. So it should be at least partially separate from the visual representation and a little bit more of tapping into the semantic representation. And we found that these object representations had reliable semantic content by this measure in CA1, in parahippocampal cortex, and in perirhinal cortex. And then if you do a whole brain version of this analysis, you see, a wide swath of higher order visual areas that seem to have this semantic content. Okay, so now, are these representations stable over time, over the course of these 30 sessions, or are they changing over time? So we did this in the regions that came up in that semantic analysis. We also did it in V1 as a kind of control region. And so these plots show an object's similarity to itself over time. So the negative slopes here mean that the objects are tending to change, the representations are tending to change over time. So each of these lines is a participant, and then the bold lines are the average. And we find in parahippocampal cortex especially, but also in CA1 and perirhinal cortex, that there is some drift in these object representations across the sessions. Not in V1, which I find reassuring, because there's a lot of drift in the brain, and maybe even in V1 in some cases, but for this type of representation, early sensory cortex seems to be stable. And here's the whole brain version of that. So it's these more anterior areas within these semantic regions that are showing this drift over the 30 sessions. So we see this drift over long periods of time, but we don't know what's causing it, right? So it could just be like endogenous change that has nothing to do with what you're experiencing in the world. It could be a result of learning. It's hard for us to figure that out from that data. But here we're going to look at a more rapid timescale of learning, so learning within one of these sessions. And here we might be able to say something more about the source of the change. So what we're going to ask here is whether the statistics of object co-occurrences in the scenes that you're seeing in a session are tweaking the way that you're representing these objects immediately in that session. And the way we do this is to calculate clusters of item co-occurrence in the first half. So just based on the co-occurrence in these scenes themselves. So this is a metric that captures just like have the objects actually been seen on the same image, but also do these objects tend to co-occur with other objects that are seen together. And then we ask whether that co-occurrence structure predicts the neural representational similarity in the second half of the session. And we have a very strong control here, which is to do the reverse. So to ask whether the co-occurrence statistics in the second half of the session predict the neural similarity in the first half. So that should not be true, or what we're interested in here is the causal influence of the statistics on the neural response. So we're going to make sure that anything we look at is the first thing and not the second thing. Okay. So what we find is that recent structure of the object co-occurrences predicts representational similarity reliably in parahippocampal cortex and marginally in CA1. In the Searchlight version of this, we found hotspots in anterior PPA, and then here's anterior PPA, and then a more anterior and medial area that falls within that parahippocampal cortex ROI that we've been using. Okay, and we find that the more the recent structure mismatches the longer-term structure of the NSD object co-occurrences, the more representational change we see. So in other words, the brain is doing more updating when there is more updating to do, which makes sense. So parahippocampal cortex seems to contain these semantic representations of the objects in these scenes that's rapidly shifting according to the recent statistics of the environment. This builds on findings that parahippocampal cortex represents long-term associations among visual objects. I think it's very consistent with that work. Here we're showing that there's kind of these rapid online tweaks to those representations. CA1 was unique within the hippocampus for containing the semantic representations, which is very interesting to us, and also showed some hints of that rapid learning, which we think is generally consistent with the way we're thinking about that region. And yeah, together with the long timescale drift findings, the results suggest a kind of a gradient of plasticity across semantic regions. And so this is evidence for this kind of bigger picture macro scale organization that we've been thinking about, where it's not just hippocampus versus cortex. There might actually be a kind of a continuum of increasing speed of learning as you go up this hierarchy, and potentially increasing overlap in representations. So then we wanted to ask, is there some sense in which this arrangement of things is optimal? Is there a reason the brain might have set things up this way? And one way to get at this is to just ask what would happen if a deep network was allowed to optimize these properties. So what would happen if it was allowed to learn each layer's learning rates and sparsity? So here we took this meta-learning approach to let networks with different architectures learn layer-wise sparsity parameters and learning rates. Okay, so each projection from one layer to the next is going to have a learning rate. That's a parameter that we're going to meta-learn, and then each layer has a parameter that controls its sparsity, how many units can be active, and that's also going to be learnable. And we tried this across different data sets and ConvNets and just fully connected networks, and the story is the same across all of these different instances. And this is the meta-learning approach that we are using. It's adapted from prior work, but we're going to concurrently learn the weights of the network alternating with the updates to these hyperparameters. And what we find is that this meta-learning approach results in better performance. And interestingly, more efficient representations overall. So this is just like averaged across the whole model. So it's choosing lower activity overall when it does this. But what we're really interested in here is this hierarchy. So what we find is that layers higher up in the hierarchy choose to be sparser. So this is the time course of this, and this is like the final state of it. And they choose to be faster learning. So as you go up, the layers are learning faster and faster. This is very cool. We're excited about this non-monotonic thing that's happening at the beginning here. So it seems like the earliest layers actually start off with higher plasticity, but then that goes away over time. It's kind of like a developmental critical period that just kind of falls out of the optimization process. So that was kind of what happens across a serial hierarchy, but we're also interested in what happens across two parallel pathways, because that's what we've been looking at in the hippocampus. And so here we took an architecture where we have a kind of input, deep serial network, and then we split off into two layers with two different sizes. So nothing else different about them to start with, other than that they're different in size. And what we find here is that the model chooses to assign the larger layer to be sparser and faster than the smaller layer. So yeah, sparser and faster. And that's very exciting to us because that corresponds nicely with how we think about the trisynaptic pathway, and in particular dentate gyrus, which is larger and is sparser and faster. So there might be some sense in which this is also this is kind of an optimal arrangement. Okay, so meta-learning, layer-wise sparsity, and learning rates results in brain-like macro organization, which we think suggests that this organization might have been optimized for behavior. This, this could play out on evolutionary timescales, maybe on developmental timescales. So we're kind of like agnostic about how the brain might be implementing this, or it could be multiple things. But it's exciting to us that this kind of thing falls out of an optimization process. Okay. So those last few projects were about the power of the feed-forward learning system. And in the last few minutes here, I want to talk about the reverse process. So for information that we think really is first dependent on the hippocampus, how is it ultimately then integrated into those existing cortical networks? And then here we get into this story of offline replay and sleep, and how might it be that the hippocampus can help to carefully establish new information into cortical networks. So I'm just going to tell you about one of the projects we're doing in the space. Half of our lab is doing sleep work, but I just want to give you a sense of the type of thing we're thinking about in this domain. So here we're going to ask how sleep and replay might serve to integrate hippocampal and cortical learning. So we think that replay during sleep is part of the answer to how this integration happens. We have a model also from Daria over there, so you can also bug him about this one, where we showed that that autonomous hippocampal cortical replay during simulated sleep that interleaves reactivation of new hippocampally dependent and old cortically consolidated information supports this kind of graceful knowledge integration. There's other theoretical frameworks that have proposed something like this, where new information, especially that's inconsistent with your prior knowledge, requires interleaved training, maybe interleaved offline replay of old and new information. So we wanted to try to test this idea. So does interleaved reactivation during sleep help with this integration process? So this is an empirical sleep study led by Bryn Sherman. who used these satellite stimuli that I showed you earlier. She had people do a prior knowledge building phase where they learned about categories of these objects over several sessions. So we're hoping, they know this stuff really well, and we give them time to consolidate. We give them a couple weeks, and then we bring them back into the lab and show them new pieces of the hierarchical structure, so new exemplars from a different part of the category hierarchy. And we're interested in how they integrate those new exemplars into their existing knowledge of the category structure. So people are learning about the parts of the satellites. They're also hearing the spoken names of these satellites as they're learning about them. And then while they take an afternoon nap in the lab, so they learn the new exemplars, and then they take a nap right after that in our sleep lab, and we're going to play the names of the satellites quietly during sleep at the right moments to try to encourage them to reactivate the associated satellites. So this is the targeted memory reactivation technique, which, if you haven't heard of it before, seems kind of science fiction-y, but it's like amazing and works really quite well and allows us to have some causal influence over what's happening with replay in human sleep. So we use this real-time protocol that was developed in this paper in the lab by Liz Seifert, but it's based on other literature that has shown that it's important to cue at really the right moments when we know that endogenous replay can occur, so the peaks of slow oscillations. We also avoid spindle refractory periods. Here's an example of the kind of evoked response in the brain to one of these cues. So the sounds are making it into the brain while people are sleeping, but they don't report hearing anything. So for each participant, we have 3 queuing conditions. For one of the categories, we're going to queue the old and the new items in interleaved order during sleep. For the second category, we only queue the new items. And for the third category, we leave it alone, no queuing. And then at the very end, we do this name sorting task where we give participants all of the names and we ask them to arrange them in this 2-dimensional space, however they feel like these things should group together. And then the question is, will participants group the satellites by the context in which they learned them, meaning 2 weeks ago versus today, or will they integrate the new exemplars into the category structure and sort more by the category structure? And what we find is that in all of the conditions, there's an effect of both. So people sort based both on the category structure and on when they learned the exemplars. But interleaved queuing leads participants to group the old and new exemplars based on category structure more, right? So the category structure becomes a more important factor for their grouping than this contextual structure. So that's evidence that interleaved reactivation during sleep indeed is promoting that integration of the new and existing category knowledge. I thought I would just put up a slide with a few of the things that I couldn't fit in today, just in case people want to come find me and chat about these things. So we have other TMR studies in the lab, one that shows that replay results in transformation of object memories as opposed to simple strengthening. Another one that TMR results in an abstracted form of memory that allows you to generalize. So we have a transfer learning paradigm. So replay can allow you to generalize to a completely new category structure or set of category exemplars if the structure is matching. And then we also have a model of the detailed characteristics of rodent replay, also some human replay, but much more focused on rodent replay findings. That's a kind of a memory-focused account that we're thinking of as an alternative to the reinforcement learning-focused account that has become very popular in that literature. Okay, summing up. So the proposal is that the hippocampus may contain these distributed representations that complement the classic pattern-separated representations, and that these are very useful for a variety of rapid, novel learning situations that require integration across experiences. But when learning involves tweaks to your existing knowledge, we think that fast learning neocortical areas are going to likely play a more direct role. And then we're interested in this possibility that there's this kind of optimized continuum of learning speed and sparsity running from early sensory areas up into the hippocampus. And then finally, interleaved reactivation of new and old information during sleep, we think might be crucial for understanding how we integrate new hippocampally dependent memories into existing cortical knowledge. Okay, I'll stop there. Thank you so much. Okay, I think there's microphone runners. I see somebody over there. Do I choose? Okay, yeah. Over here.
Thank you for the very interesting talk. I wanted to ask something in a different perspective, from a prediction perspective. If you are in a continuously evolving environment and you have to do predictions based on learning, and this environment is fastly evolving, do you rely on more on a monosynaptic pathway and more recent examples, or you rely more on a trisynaptic pathway and you build a representation of the whole statistical distribution? So which one is gonna be activated more? So that was my question.
Yeah, the way we're thinking about it right now is that you kind of have learning turned on everywhere all the time. So you have cortical learning turned on that's tweaking things all the time to the extent that it's able to incorporate, a small adjustment from your existing predictions about what was going to happen. And at the same time, you're learning in these two hippocampal pathways in parallel these different kinds of representations. One that's going to be sensitive to the statistics across the new experiences, and then the other that is going to just take snapshots of kind of each of your experiences. And then we're relying on some kind of smart deployment process later. So we're exploring now this idea that the prefrontal cortex might be able to help you figure out, okay, now I need to make a decision. What should I use, right? Right. Yes. So the question is, could the uncertainty of the environment affect how you deploy these things? I mean, I feel like Kate has a meta-learning, where is she? Wiesenbaum has a meta-learning project, well, you can go talk to her, but I think she has a simulation that kind of does that, where you learn, or a theoretical paper. You learn based on the statistics of your environment, kind of what you should do. I think there's other reinforcement learning literature that takes that kind of approach. We haven't done that yet, but you can imagine that like a mixture of experts type of approach could do that kind of thing. So say like it's not just about the task, which representation you should deploy, but also you might learn that, in a more uncertain environment, maybe it's more useful to use a statistical learning system. So that seems very possible. We haven't done that yet.
Wow, thank you so much for an awesome talk. There was a lot of really cool results in there. I was wondering, so I'm kind of interested in cognitive fatigue, and there's this, Mayank Agarwal with Nathaniel and John Cohen did, this idea that you can learn more efficiently in RL tasks from replay, and so you can find like an optimal stopping time to stop sampling the environment and instead replay. And I was kind of wondering if that could interface with your kind of PFC, meta learning between these systems. I don't have it that connected in my head, but I was wondering if that's something you could imagine drawing some connection to.
Yeah, I love that. I didn't have that connected in my head either. And I think that's a cool idea. So like it's like not just about the hippocampal learning or the way you want to deploy your hippocampal representations, but about kind of how do you decide when to allow the integration between systems, right?
Right. So learning being active process and you engage, but also decide how to stop it.
Yes, I love that idea.
I don't think about that, but thank
will
you.
do
It's
it.
really, really, really cool ideas you put forward.
Cool, thank you. Is this the online world?
Yeah, nothing new here.
Up there?
Thanks for a really thought-provoking talk. So I had a similar question, or along a similar line. I was really intrigued about that idea of the MPFC and the mixture of experts, and I wondered to what extent one's goal or future expectations could likewise sort of shape that relative balance.
Yeah, that's exactly the idea. So you could frame it as task. I said task, but I think you could call that a goal. So if your goal is to generalize across exemplars, then So the idea is that this system is sensitive to what you're trying to do, what you want to do in this environment, and then deploying the right representation. Yeah.
Cool, that's what I was thinking too.
Yeah.
And do you think that extends to replay too? So that.
Oh, you guys are making me find all the connections across. Okay, does it extend to replay? The idea that your goal. Okay, so yeah, that's interesting. Our replay model that I mentioned, the idea of the model is that. It's surprising the range of characteristics of replay you can explain from a simple, dumb, memory-focused perspective without appealing to goals. So there's quite a lot of forward and backward replay and differences between wake and rest and all kinds of overrepresentation of reward, all these things that you would kind of naturally interpret as being about value learning or about trying to plan to achieve a goal, and we show that actually simple memory, contextual memory dynamics explain a lot of that. So I suppose in that space, we've been thinking that that endogenous replay might be a little bit dumber than you'd think, like a little bit more thinking about just building a general statistical representation of use for many possible future tasks instead of optimizing for the particular task that you're in right now. So, but of course, it's all these things interact, and I could imagine that there's some kinds of replay in the moment that you might expect to be more task dependent. But our way of thinking about replay is that it's trying to build up something general for use in many situations, less optimized. Yeah.
Hi, fantastic talk, thanks. I was wondering about, there's this rather old line of work looking at pair coding in inferotemporal cortex.
Yeah.
So I was curious how that fits into your story, because then that's actually sort of like really sensory or like category selective sensory regions, which ultimately basically also start representing the statistical regularities. Is that something sort of that happens at the very end of this? Or?
Yeah, so the old Miyashita studies, that was why we ran that first experiment I showed with the pairs of objects in the sequence. That was why we ran that experiment, because we wanted to think about how this might play out across brain regions. If we could look with fMRI at a lot of regions at the same time, where would we see that? On what time scale would we see those kinds of pair coding effects? And so the Miyashita studies were very focused on, they called it IT, but I think it was perirhinal cortex. And there were some of the studies in that line of work actually looked at kind of where does the pair coding emerge earliest and where does it emerge later, and they found that it was more anterior areas that showed the emergence first, but the time scale is like days, maybe weeks, not minutes, right? And so the way we're thinking about it is that's exactly the kind of medium range, that fits into this gradient story of MTL cortex to anterior neocortical sensory areas. But that if we're going to try to do this on the timescale of minutes, which is not what they were doing in those studies. You're going to have to go into the hippocampus. But we think it's very consistent with that general way of thinking about things. People can always come find me at the, I'll be here through Thursday. Excited to chat with people at the conference.
Excellent. Let's thank Anna again.